The human gastrointestinal tract is a continuous tube extending from the esophagus to the anal canal. It is an internal biological barrier engineered to withstand severe mechanical friction, synthesize corrosive acids, and dissolve dietary fats without destroying itself. We will track 3 specific processes: the histological blueprint of the tract wall, the biophysics of gastric acid secretion, and the fluid dynamics of hepatobiliary detergents. We start with the architecture. The entire tract maintains a conserved 4-layer structural blueprint. Building inward from the exterior, we have the serosa or adventitia, the muscularis externa, the submucosa, and finally the mucosa in direct contact with the lumen. The innermost mucosa is further subdivided into 3 parts: an epithelial lining, a lamina propria containing gut-associated lymphoid tissue, and a thin, smooth muscle layer called the muscularis mucosae. Right beneath that sits the submucosa, a dense connective tissue layer holding larger blood vessels and the neural wiring of Meissner's plexus. At the outermost border, you must distinguish between the serosa and the adventitia. Intraperitoneal organs, like the stomach, are wrapped in a lubricating serosa, while retroperitoneal organs, like the esophagus, are anchored directly to surrounding tissues by a fibrous adventitia. This universal 4-layer layout is highly adaptable. Targeted regional modifications allow the body to survive the hostile chemical processes required for digestion. A perfect example of this regional adaptation occurs right as food exits the stomach. If you look at a histological slide and see glands packed into the submucosa, you are looking exclusively at the duodenum. These are Brunner's glands. Their sole purpose is to secrete a highly alkaline mucus with a pH of 8.2 to 9.3 to rapidly neutralize the incoming wave of lethal gastric chyme before it burns through the small intestine. We see another extreme adaptation at the gastroesophageal junction. Here, the epithelium abruptly transitions from a thick, stratified squamous layer built to resist the physical abrasion of swallowing, directly into a single layer of secretory columnar cells built for chemical processing. When this junction fails during chronic acid reflux, gastric juice forces the squamous tissue of the lower esophagus to adapt. The tissue undergoes intestinal metaplasia, transforming into columnar cells with goblet cells. This condition, Barrett's esophagus, is a known premalignant lesion. Observing how microscopic architecture reacts to localized stress allows us to track the progression of pathology and predict fatal outcomes like esophageal adenocarcinoma long before a tumor forms. Inside the stomach, the physiological challenge shifts from mechanical protection to active chemical generation. The stomach must synthesize hydrochloric acid at a pH of 1.0, pumping protons against a million-fold concentration gradient. This table outlines the 6 specialized cell types within the gastric mucosa that make this extreme environment possible. Note the strict separation of cellular duties on the left and their specific secretory products on the right. Looking at the stimulating signals column, you see a division in how these cells communicate. G cells act endocrinely, releasing gastrin directly into the bloodstream. ECL and D cells act paracrinely, signaling only their immediate cellular neighbors. D cells specifically trigger a negative feedback loop to shut down acid production when luminal pH drops below 1.5. The primary engine of the stomach is the parietal cell, which is responsible for 2 exact outputs: synthesizing hydrochloric acid to denature proteins and secreting intrinsic factor to bind vitamin B12. The stomach prevents autodigestion by strictly compartmentalizing these chemical responsibilities, the mucus barrier, acid generation, and neuroendocrine signaling, into entirely separate cell lines. To understand how the parietal cell actually achieves that million-fold gradient, we have to look at its specific intracellular ion transport machinery. This diagram illustrates the mechanics of acid secretion, starting intracellularly. Carbonic anhydrase catalyzes the hydration of carbon dioxide in water, yielding carbonic acid. This immediately dissociates into a free proton and a bicarbonate ion. That newly generated proton is actively extruded across the apical membrane into the gastric lumen by the proton pump, the H+K+ ATPase. This pump consumes ATP to force the proton out while importing potassium. Chloride then follows passively through apical channels to form hydrochloric acid. Meanwhile, the remaining bicarbonate ion is pushed out of the basolateral membrane into the venous bloodstream by the AE1 exchanger. This specific efflux of bicarbonate raises the pH of the postprandial venous blood, creating what is known as the alkaline tide. If these parietal cells are destroyed by an autoimmune response, acid production fails. But more critically, the loss of intrinsic factor means vitamin B12 cannot be absorbed later down the line in the terminal ileum. Without B12, the body cannot sustain proper erythropoiesis or maintain the myelin sheaths of the spinal cord. This specific microscopic pump failure triggers the systemic collapse seen in pernicious anemia, from megaloblastic blood cells to the demyelination of the posterior columns. Running millions of proton pumps requires massive ATP expenditure. To prevent wasteful baseline secretion, the parietal cell is held under strict neuroendocrine regulation. This flowchart tracks that regulation. Acid secretion is controlled by 3 stimulatory receptors, M3, CCK2, and H2, and 1 primary inhibitory receptor, SST2. All 4 of these inputs converge directly onto the proton pump. They dictate pump activity through 2 distinct secondary messenger pathways. The H2 receptor utilizes cyclic AMP to activate protein kinase A. In contrast, the M3 and CCK2 receptors trigger an increase in intracellular calcium to drive pump activation. These receptor pathways are engaged sequentially across the 3 phases of digestion: cephalic stimulation, triggered by the sight and smell of food, the gastric phase, driven by mechanical distension, and the intestinal phase, governed by duodenal feedback. This receptor architecture dictates our pharmacological targets. H2 receptor antagonists like cimetidine block just one pathway, reducing overall output. However, proton pump inhibitors like omeprazole covalently bind and irreversibly block the ATPase itself. By memorizing these distinct receptor pathways, clinicians can completely bypass complex, multi-organ feedback loops and definitively shut down acid production at its final common pathway. When chyme enters the duodenum, a new biophysical challenge emerges. Dietary lipids are hydrophobic. In the watery environment of the intestinal lumen, they coalesce into massive droplets, presenting almost zero surface area to water-soluble pancreatic lipase. The liver solves this by synthesizing bile salts. These are amphipathic detergents, equipped with a hydrophobic steroid core to bind lipids and a hydrophilic tail to interact with the aqueous fluid. Lipid digestion requires 2 distinct interventions from these salts. First is emulsification. Mechanical churning physically breaks large fat globules apart. The bile salts immediately wedge themselves at the air-water interface, keeping the droplets small and drastically increasing the surface area for lipase to attack. Once lipase hydrolyzes the fat, the resulting free fatty acids remain hydrophobic. This brings us to the second phase, micelle formation. When luminal bile salts reach the critical micellar concentration, they spontaneously aggregate, forming highly stable spheres that safely transport the lipids through the unstirred water layer. Bile acts as a vital biophysical bridge, temporarily altering the physical state of lipids so that water-soluble enzymes can access and process hydrophobic fuel within the aqueous intestinal fluid. This system faces a severe supply deficit. Digesting a single fatty meal requires up to 30 grams of bile salts, yet the human body only maintains a total pool of about 4 grams. The math is balanced by enterohepatic circulation. The total bile pool is recycled multiple times per meal. After lipids diffuse into enterocytes, over 95% of free bile salts continue to the terminal ileum, actively scavenged and reabsorbed by the ASBT cotransporter. Once reabsorbed, these bile salts bind to albumin and return directly to the liver via the portal venous circulation, ready to be resecreted. If a patient requires surgical resection of the terminal ileum, for example in Crohn's disease, those ASBT transporters are removed entirely. The liver cannot synthesize new bile acids fast enough to replace the loss. Micelle formation fails, and unabsorbed fat is excreted as foul-smelling phlotolyses, a condition called steatorrhea. Removing the downstream absorption endpoint entirely starves the upstream function of the liver, proving that the gastrointestinal tract relies on a total closed-loop structural interdependence. Let's map these biophysics onto a single chronological timeline. The journey begins with fat ingestion. This list outlines the process. When fatty acids enter the duodenum, they trigger eye cells to release cholecystokinin. This hormone commands the gallbladder to contract, deploying bile. Amphipathic salts coat the lipid droplets to stabilize a coarse emulsion, allowing pancreatic lipase to cleave the triglycerides into free fatty acids and monoglycerides. These hydrophobic products are immediately sequestered into the core of bile salt micelles. The micelles ferry them across the unstirred water layer, where the lipids passively diffuse into the enterocyte. Now empty, the free bile salts continue their descent to the terminal ileum, where they are actively reabsorbed by ASBT transporters and returned to the liver via the portal vein. This synchronized assembly line relies on exact timing between hormonal triggers and biophysical state changes at the cellular level. Let's lock in 4 high-yield details for your exam. Trap number 1, location. Brunner's glands are strictly located in the submucosa of the duodenum, never the mucosa. Trap 2, receptor pathways. The H2 receptor elevates intracellular cyclic AMP, whereas the M3 and CCK2 receptors rely strictly on intracellular calcium. Trap 3, spatial dissociation. Intrinsic factor is secreted by parietal cells in the stomach, but its actual physiological action, vitamin B12 absorption, only occurs in the terminal ileum. Trap 4, biophysical chronology. Biosalt emulsification must occur before lipase digestion, and micelle formation only occurs after lipase has done its work. To cement these concepts, take 10 minutes to actively draw out the parietal cell machinery and the GI wall cross-section as mapped in your compendium. Mastering these microscopic biophysical details gives you the tools to diagnose and reverse life-threatening systemic pathologies exactly where they start.